Hello, everyone, and welcome to today's webinar. This webinar is hosted by Jen and sponsored by Sanova Analytical. Today, we will be discussing a strategy for reproducible protein quantitation in gene therapies using Ion Mobility Spectrometry. Thank you so much for joining us. I'm Uduak Thomas, Senior Editor with Jen, and it's my pleasure to be your host for today's webinar. You'll be hearing from three speakers today. Dr. Mark Fielden, Senior VP Non-Clinical Development at Kate Therapeutics, Jason Apter, CEO of Sanova Analytical, and David Jones, VP of Technical Solutions at Sanova Analytical. Let's start off with some background on today's topic. Accurate protein expression quantification is critical for gene therapy development. However, measuring protein expression reproducibly is a challenge for many traditional assays, and this is due to limitations in sensitivity and selectivity. For today's gene therapy developer, Ion Mobility Spectrometry, or IMS, delivers the sensitivity and selectivity needed to accurately identify and quantify protein products across complex biological matrices. Now, compared to traditional technologies, IMS reliably distinguishes target protein products from similar endogenous compounds and detects therapeutic proteins expressed at low levels. It is also adept at navigating blood and tissue samples, where such complex environments can add variability to chromatographic readouts. So in this Gen webinar, our panelists will discuss strategies for overcoming challenges in protein quantitation using ion mobility spectrometry from early R&D through preclinical and clinical trials for gene therapies. So what are some of the key takeaways from today's webinar? Well, you'll learn how the technology can resolve isomer differentiation and low abundance protein challenges. You'll hear how it can increase confidence in target protein expression quantification results. And you'll hear about an accelerated and scalable strategy for gene therapy development. Today's webinar will be followed by a live Q&A. At any point during the event, if you have a question, you can enter it in the Ask a Question panel on your screen. All right, let's get started.
Thanks, Uduak, for the kind introduction, and thanks, Mark and David, for joining us on today. Just a bit of context, what we were hoping to accomplish in today's discussion is a few things. First of all, is really have a discussion on the role of quantitative proteomics in supporting gene therapy development. Secondly, we're going to dive into the inherent challenges in quantitative proteomics as it relates to gene therapy development as well. And then we're going to take you through an overview of some traditional techniques that are used in quantitative proteomics and some of the trade-offs amongst the different approaches. And then finally, we'll introduce a new bioanalytical solution that we're excited about that could offer and provide fewer trade-offs than traditional bioanalytical solutions. To start the discussion, we wanted to set the context of why this is important. I don't think I need to talk to anyone that's joined us on this webinar to know that gene therapies are a very exciting field of drug development. And it's been very interesting to watch the progression of the pipeline in gene therapies. And you can see from the chart that we've now come to a point where we've got over 2,000 therapies in the pipeline, both in the preclinical stages and in the clinical stages. And an interesting thing that we're seeing is that as we move forward, while the number of target or assets in the pipeline has not grown over the past few years, the progression of assets from preclinical to clinical is actually starting to grow a lot more, which is very exciting. And this lends itself to the need for protein quantitation and really our understanding of the various different applications that it's used for in gene therapy development. First off, evaluating therapeutic efficacy and biodistribution. Secondly, understanding post-translational modifications. Third, work in biomarker discovery. Fourth, understanding therapeutic mechanisms of action. And fifth, optimizing gene therapy design. I'd like to take the time, Mark, to pull you in and talk about your thoughts, having been in the sector now for a long time and really dealing with a lot of these applications, kind of your thinking with respect to the market itself and how critical protein quantitation is to the development of gene therapies.
Yeah, thanks, Jason. I come from a gene therapy background, working at Kate Therapeutics. There's obviously a lot of excitement about gene therapy. There's still a lot of improvements to be had. I think Kate Therapeutics was a good example of how we could successfully apply some new technology to solve some really important problems, specifically the delivery problem, the problem of delivering therapeutic proteins back to tissues. The other attractiveness of gene therapy is that they're typically one and done treatments. Single injection could last years, if not decades, particularly for terminally differentiated tissues like CNS, muscle, or heart. I think durability for liver might be a little bit more challenging. The other benefit is that we're working on genetically validated targets. The targets are typically mutated, and the beauty of gene therapy is to replace that mutated gene with a wild type healthy copy of the gene. So just having that target validation is reassuring for development. And because of that, we typically see large effect sizes, typically life-changing efficacy. And I think there's some good examples in the field. Zolgensma is one of the early examples. That's a very serious disease that affects young children, spinal muscular atrophy. And we're just seeing an incredible degree of efficacy in these boys. There are still some safety challenges, but like I said, there's a lot of new technology like engineered capsids that are getting better, that allow for better efficacy and better safety. In terms of the relevance of quantitative proteomics, there's still some catching up in the field that proteomics has to do relative to some more established quantitative techniques like quantitative PCR or ddPCR that's been around for decades and is really good at quantitating nucleic acids, which is important for gene therapy. But typically, the therapeutic cargo for a gene therapy is a protein, usually replacing the missing or broken protein. And so we really need to address where that protein gets expressed and how much is there, both preclinically and clinically. And in fact, understanding the biodistribution of the protein or the therapeutic cargo is a regulatory requirement, typically evaluated in multiple species, small animals like a mouse or a large animal like a non-human primate. And this requirement to evaluate biodistribution is detailed in a guidance document, ICHS 12, which is still in draft form. But I know a lot of sponsors are already following those guidelines quite tightly. And this is important clinically as well, so that we can tie back the levels of the protein in the target tissues back to the preclinical species to understand if we're actually expressing enough therapeutic cargo in patients, and should we expect clinical efficacy at those levels of the protein. Some of these other needs affect not just gene therapy, but other modalities as well. Post-translational modifications, less common for gene therapy, but really common for kinase inhibitors, for instance, where you're measuring changes in phosphorylation levels. Biomarker discovery is really important for gene therapy. We don't have the luxury of taking large tissue samples, particularly for CNS, where biopsies are just not going to happen. But biomarker discovery can be had in more accessible fluids like CSF, for instance, or plasma, of course.
Thanks, Mark. I mean, it is exciting, and it does seem that the need for continuing to improve the tools and techniques used for quantitative proteomics would help the development and growth of the gene therapy pipeline for sure. Just shifting over, David, you've been in the industry for many years. Your thoughts on just the overall market and some of the needs that you've seen from your perspective.
Well, I mean, the market is certainly, in my long career, I've seen a number of modalities change, going back to the early days of proteomics itself, when you in fact were injecting the proteomic drug into the patient and following it. This is in gene therapy. We're really talking about protein expression, and the expression of proteins. We've had a lot of traditional methods that have been used. Most of those involve molecular biology techniques, and what I've seen in my career, really accelerating over the last 15 years, 15 to 10 years, is the use of mass spectrometry to not necessarily completely supplant, but to elaborate on the traditional methods that have been used. And I know that in our work with Mark in the past, we've kind of worked to put those methods together and just try to utilize the strengths of each. But again, this technology is being so exciting and being so important, and really affecting what can be minor proteomic changes. You might even go down to a level of a single nucleotide polymorphism. You really do need to have a highly accurate and precise understanding of exactly what's going on.
Well, that's a good segue, David. As we talk about, we all know it's an exciting field in curative therapies, which is exciting that gene therapy is now, I think the pipeline is now progressing, not just, basically oncology focused, but there's a lot of different indications, rare disease and others. So it is an exciting time. But as you get into then the issues and challenges with respect to quantitative proteomics, and what we've kind of thought through is, as you look at some of these issues that have or challenges that have provided and we still need to resolve, we kind of simplified it into 5 key areas. First of all, with certain therapeutics, you're really looking at low abundance proteins, and the ability to see them at very low sensitivity levels is important. The other area and challenge that we've seen commonly is really understanding the difference between the endogenous compounds versus therapeutic proteins. A third that we're very familiar with is the complexity of biological matrices. And Mark, as you talked about new delivery systems, I think as we get delivery into different areas of the body and the different organoids, the complexity of these samples is getting more and more challenging. And then some of the other things, with limited sample volume and the continued need for accuracy and reproducibility, it's clear that there's some common challenges that have always revolved around and have helped the field evolve. But it'd be interesting, Mark, to hear your opinion and perspective on with the advent and even some of the innovations and trajectory of where gene therapy technology has gone to. What's your view kind of on the impact it's had on some of these common challenges would be interesting to hear.
Mm-hmm. Yeah, I think the accuracy and reproducibility is probably at the front of my mind. It's sort of table stakes for any quantitative approach, but it's particularly challenging with proteins. Most of the proteins that we're dealing with therapeutically are not that high abundance. And so getting accuracy and enough precision or reproducibility at that low end of the range is typically the problem. And with any immunoassay-based approach, you're sort of limited in the reagents or the antibodies you have. This problem around low abundance proteins also touches on limited sample volume. For preclinical studies, it's typically not a problem to get enough tissue. You can obtain that, through necropsy. However, clinically, we're often dealing with very small biopsies. Could be an endomyocardial biopsy from the heart. Could be a biopsy in the leg to get some skeletal muscle tissue, or even just CSF fluid, for instance. And when you have very little sample volume, picking up those low abundance proteins can be particularly challenging. Even some of that biopsy sample needs to be, used for nucleic acid isolation, even for histology. So there's just not a lot of sample to go around. And I think the other challenge here is what was alluded to before, differentiating endogenous versus therapeutic proteins. We were actually dealing with this at Kate more recently, where, in the context of a monkey study, non-human primate, where the gene therapy is expressing a wild type human protein, and we're trying to differentiate that from the endogenous non-human primate protein, which has extremely high amino acid similarity. So no antibody-based approach is ever going to be able to differentiate that. So and that's going to be really important for gene editing as well, where we're often just dealing with a single amino acid change.
Thanks, Mark. That makes a lot of sense, and it does lend itself to the fact that, as the excitement and I guess the promise of some of these new therapies also amplifies some of the existing challenges that we have to go with. David, you've kind of watched this progression throughout as well. I mean, your perspective on some of the common challenges and how they're evolving now as the therapeutic modalities evolve.
Yeah, so I mean, the big changes are, as Mark alluded to, are these very, very small changes in what are not abundant proteins, and these changes can be almost trivial, and sometimes you have to differentiate between, in a case where there's a single amino acid substitution. They may be close in mass, or as the example that Mark gave of working with a trans species. If you're working to put a human protein or generate a human protein in a test animal, you have the problem that the test animal may have a lot of homologies. There are many conserved sequences across proteins. They don't vary dramatically from between species and species. Some of them are found through the entire range of the kingdoms of living systems. And so it becomes a challenge. You might be looking for a very narrow region that you have to differentiate. And depending on the technique that you use, and certainly the instrumentation you use, you're going to face a plethora of problems. And that's why, we've been working to embrace even more and more modern analytical techniques, again, centered, chiefly in mass spectrometry. But it's the tools need to evolve with the technology. That's
You segue perfectly as always, David. And I think that gets into kind of, as you said, really as the modalities continue to evolve, as we get better technology, the need to support those with quantitative tools needs to evolve with that. So very clear. I think that's a good kind of transition into maybe a high level discussion on some of the traditional approaches that have been used to quantify proteins, and really understanding some of the pros, why we're going in these directions in certain areas, but also the cons and what are the limitations that certain approaches may have. Mark, I'd love to hear your perspective, particularly on the immunoassay-based approaches that have been really used throughout to do protein quantitation.
Yeah, I can certainly speak to those on the left side of the slide where most of my experience is. I mean, the benefits of immunoassays, whether it's Western blots, capillary electrophoresis, or even ELISAs, is that they're generally very accessible to most molecular biologists. We grew up with these tools. We use them in grad school. They're generally more affordable, and so most labs have that capability. The challenges come in when you start thinking about the actual reagents you have to use. You need an antibody to detect a protein, and your assay is really going to be governed by how good your antibody is. Is it sensitive enough? Does it have a high affinity for the target antigen? Is it specific for the target antigen? And this can be a challenge when you're trying to differentiate human wild type protein from the endogenous monkey protein, as I alluded to in the previous slide. in terms of precision reproducibility, they can tend to be pretty good, but it just depends on what the needs are and what sort of level of precision you need. At the low end of the range, precision's it's not going to be as favorable, and that might be a problem for your ability to accurately define what the therapeutic protein levels are. You need a loading control sometimes to control the amount of input protein. So there's essentially another assay you need to optimize for the loading control, which might be, beta tubulin or some housekeeping gene that's expressed in your sample. There's other challenges like cross-reactivity of your antibodies, not just your primary antibody, but your secondary as well. So there's a long laundry list of pitfalls that you might come across. But generally speaking, I think they've shown their worth, and most molecular biology labs have had some good success with some of these approaches.
No, it makes sense. You're bringing me back to my days in the tool space where we were designing antibodies and even designing multiplex assays, and that was always the challenge, is making sure that you had very solid, good selective antibody pairs and validated pairs, and so and as well the rest of the reagents that go with it. So it's a blast from the past for myself. Maybe David, knowing you've spent a lot of your career on the mass spec side of the space. It'd be interesting to hear your perspective on how mass spectrometry really has evolved over the years to really become a viable alternative to some of the traditional immunoassay-based approaches.
Yeah, so mass spectrometry is actually over a century old. It's generated tons of Nobel Prizes over the years, but really the interest in the use of mass spectrometry for biological purposes really started about 1980 with the invention of a soft ionization technique called ESI, electrospray ionization. And that technique has revolutionized the way that we can see biological molecules and small molecules. What it offers is that it's the same every time. Okay, I'm within some limitations, but basically you have the advantage of being able to synthesize heavy stable isotope labeled reference standards. And so you can get a real good sense of the reproducibility. And if there is a change of some type, you will pick it up. And so we show here sort of a few relatively modern mass specs. The quadrupole is a triple quad. It actually selects for just one particular ion. So it has some limitations in efficiency. However, because it cleans up the array of ions to limit it to a single ion, it can give very high accuracy. Okay, and you can get very high sensitivity. So those have been workhorses for around. And then as we moved into larger molecules, proteins, we became necessary to develop high resolution mass spectrometry. Quadrupole Q-trap will give you one digit after the decimal point, whereas the Orbitrap, which is shown in the lower right corner, will give you 4 areas after the decimal point. And in as much as it does that, you can now differentiate between peptide sequences within a protein with a very, very high accuracy. And the software is designed to pick on those masses, including the fragmentation masses, to tell you that you really are seeing the sequence. This is the sequence. If there is a post-translational modification, a phosphorylation, even a glycation, you can actually define that is there and that it's present. And you don't really have to rely on the viability of a capture antibody or anything else like that. It will be the same time in and time out. You do face, because you're working complex matrices, you do face some issues with, particularly if you want to do what's called data independent analysis, where you may be looking at hundreds and even thousands of proteins at the same time. You do face some issues with sensitivity. And so the more modern instruments, including those that, we've added recently, help overcome some of those limitations. So, the joke around here at Sonova is that I'm a mass spec fascist, because I think mass spec is just evolving so fast. In my career, I've seen it evolve so fast that it pretty much will rule the world in the next decades.
Thanks, David. I'd like to click into this a little bit more. As we've thought through it and as we talked, around today, there are some general trade-offs, I think, that everyone's working through when they're looking at the various different approaches. When you're looking at, first and foremost, what we heard from you, Mark, is obviously cost and ease of use. And as you said, molecular biologists have just grown up with immunoassay-based tools, so they're very well known. And these tools have also really evolved so that, you've now got automated Western blot, for example, and that makes it a lot more easier and obviously a lot more consistent by having automation in your system. The second thing that we've talked a lot about is the increasing need for selectivity. So being able to find the protein or analyte of interest in complex samples and all that good stuff. And then the sensitivity, as we've talked about with sample size, volume, and all of that good stuff, is being able to continue to drive down the lower limit of detection to really go after proteins that are very lowly abundant, or when sample sizes are small. So those three issues, and then obviously it's always been about reproducibility. You always want the results to be consistent, and the analysis to be consistent, and to be able to reproduce results through the similar assays will always be important. And then finally, it's one of the things we haven't touched on is the time to develop, right? And I'm sure that time is an area that's critical, considering that at the end of the day, I've always heard the analog is you want to, if you're going to fail, you want to fail fast, right? And progression through the clinic's important. So, Mark, I'd love to hear kind of your perspective as it comes to the approaches on those 5 key areas, and the relevance, and maybe where you see more importance as far as when trade-offs come.
Yeah, your point about the development time is important. I don't think something that we touched on. In terms of capillary electrophoresis, it's relatively high throughput versus, pouring an agarose gel, waiting for it to solidify and running for hours. So for a molecular biologist in a research lab, trying to screen antibodies and trying to figure out an assay that works is really important. And so for early stage preclinical research, optimizing an assay and getting it to work quickly is really important. And I think that's where some of these approaches really excel. But I have seen, projects evolve over time where, that fit for purpose precision was fine during early stage proof of concept studies in your mouse model. But when you start getting into development, later stage development, by that I mean GLP toxicology studies, IND enabling studies, you need a higher degree of rigor in terms of accuracy and precision. And so I have seen projects essentially transition from a Western blot platform in early stage research to a more quantitative, more sensitive mass spec based approach. Just because for whatever reason, the Western blot or the antibodies just weren't good enough to provide the degree of accuracy and precision necessary to support development, including clinical trials. So I think they're complementary. I think both approaches have their pros and cons, and you know, I think it's up to individual users to decide what's the right tool for the job.
No, that makes a lot of sense, Mark. And David, and knowing that you do quote unquote coin yourself as a mass spec fascist, any thoughts from your perspective in a balanced way, but on how you view the trade-offs, and and when you're thinking through bioanalytical solution for a particular program, how you're viewing those trade-offs.
Yeah, well, I mean, I'm not saying that that immunoassay based approaches are never going to go away. I mean, I worked on those very early in my career when we were doing radioactive things, but really when it comes down to, I think what Mark was touching on is when you're starting to get more into a development phase, right? Because even if you're going trans-species, right, you're really now at that point, you're working with an organism that might have a different proteome of that you use, say, let's say, in a mouse or a rat. If you're going to, if you're doing ocular studies and you're working in a rabbit, that's a different animal. And if you're, doing other kinds of studies in monkeys or other species, and eventually you're going to get to a human being, right? And when you get to that human being, they're going to have the proteome that you want to work with and the proteome that you want to modify. And this is the place where mass spec becomes, to my mind, essential, because you really are looking at that level at the sequences themselves. You are actually identifying exactly what the protein looks like, what its mass is. There's only a few places in proteomics that you can have, mass interference. Usually that involves leucine and isoleucine, which is generally trivial, but you can see everything. And I remember the first time I saw some of this, some of the results put out by mass spec run, in minutes, a minute time scale, where you would get back data that would say, this was stuff that in the 1950s, 60s, and maybe even the 70s, you get in 5 minutes what people spent their entire graduate careers doing. So it's a very, very powerful tool, and it's becoming more and more powerful, because right here we're looking at, at traditional mass specs, high resolution and quadrupole mass specs, but now there are a number of hybrid mass specs that actually add more dimensionality to separations of proteins. So you're really getting the thing that you really want to look at and really understand its presence and its behavior.
It makes sense, but I think that in a nice part about this discussion on this part is, I think, it's interesting, the ability to transition through various techniques, like the all-encompassing fit for purpose. But it sounds, as we progress closer to clinic, and you get into, Mark, as you said, the GLP tox and everything, the need for the accuracy, the selectivity, and the sensitivity, and the reproducibility of those really becomes accentuated, and that making that trade-off becomes important, and hopefully that's what drives more and more of the pipeline from preclinical into the clinic, frankly. I'll shift the discussion now, guys, to hopefully what a number of the folks on the call are interested in, but talking about a new bioanalytical solution that may potentially address both selectivity and sensitivity simultaneously, while also providing a speedy platform to develop methods on. David, I'd love you to take us through this approach, but again, it's the our view of ion mobility spectrometry, along with various other what I would call fringe or supplemental tools that complete the whole system, that really amplify the IMS capabilities of the past to really address some the technical issues within within quantitative proteomics. David?
Yeah, sure. So IMS stands for Ion Mobility Spectrometry. It's a technique that was originally developed in the 1950s, but it never really got integrated into mass spec until maybe the late 2000s and early 2010s. And what it does is it adds another permutation to what a molecule is. Up until now, all the mass specs have relied on electromagnetic fields, right? So you're really dealing with the ionization capability. But an ion mobility instrument is an instrument that adds the capability, another parameter, which is the shape of a molecule, or the average shape of it is. And what it involves is that you have, on a simple level, what was called drift tube IMS. You have a gas field, and it's opposed by electric field, and the gas field acts like a mast on a ship, and it propels it in one direction, and you can kind of trap ions in that case. Most of the instruments that have been developed by IMS, and there are a number of manufacturers working in the field. Most of them do have a slight, they can have a slight disadvantage in terms of sensitivity. You might lose a few ions. There is what's now called, and we're showing here in this slide, trapped ion mobility. And actually what this does is it works with an RF field and a gas flow field to actually trap ions of a specific shape and specific charge and specific mass. So you're really cutting out all the stuff you don't want to see, okay? And you can actually see thousands of things, but see them one at a time. And what it does is it increases ion efficiency. And by increasing ion efficiency, you increase sensitivity. You have the TOF analyzer, which gives you the accuracy. These things take on the order of, fractions of a second. So you can see a lot of things in a very short time. And if you look on the right side of this slide, to talk to this issue of limited sample volume, you have the ability now to work with a nanoflow. And that means instead of needing, 50 milligrams of a tissue or 10 milligrams of a tissue, you can get away with fractions of a milligram. And so you can repeat an experiment. Mark says, sometimes you have to send things out for genome analysis or other forms of analysis or biomarker analysis. So if you're looking specifically at the proteome, you can really be economical with the tissues. And so the IMS, besides increasing selectivity and specificity, right? Also, in the case of a trapped IMS, can increase the sensitivity. So you're really getting all three of those important issues addressed in one run, really, when you inject the sample. And it's really a remarkable thing. And with powerful software and sequence analysis, you're really zeroing down and really sorting through a huge mess of proteins. In this instrument, you might see 8,000 proteins in a single run, and you'll know exactly which one you're looking for and be able to tell you how much is there and what does it look like. So it's very powerful.
That makes sense. I mean, it's kind of like any technology evolution. It's the intersection of a number of different technologies. We talk about the ion trap, we talk about ion mobility spectroscopy, we talk about the TOF analyzer, and then you add on things like the ability now to do nano LC, and even what we didn't touch on is some of the the advances with sample preparation tools that a lot of the tools companies now are working on for mass spec based specific tools. you really, the ability to combine the best of the best, does absolutely provide the opportunity to, maybe address multiple trade-offs where in the past, without the integration of all of those technologies, we weren't able to address multiple trade-offs at the same time. Mark, I'd love to hear just your thoughts, having been someone that's seen this firsthand and maybe seen this up, against some of the traditional methods, your thoughts, on some of the benefits and even some of the opportunities or challenges that are still relevant to make this a viable bioanalytical solution for drug developers in gene therapy.
Yeah, I mean, in the relatively short time that I've been involved in this particular project that's taking advantage of this, the degree of sensitivity is night and day compared to immunoassay-based approaches. And really the key driver to go down this path and use IMS is the specificity and just being able to differentiate the human isoform from either the mutated isoform in your mouse or your human or the endogenous monkey isoform. So those are the two key advantages. The challenges is the development time. It's just not as rapid as an antibody-based approach. And so with appropriate planning, you can get around that and account for it. But it was absolutely critical to support progression of the program. And so we had enough advanced planning to do the development we needed.
That resonates. That's very good thoughts, Mark. So listen, I'm gonna wrap it up, guys. And then open it up to hopefully some Q&A from the audience today. But hopefully everyone's been able to glean some insight into our discussion today. I think the 4 takeaways that we wanted to leave with everybody, are first and foremost, protein quantitation is crucial really to the successful growth and advancement of the gene therapy pipeline. Secondly, there are inherent challenges in quantitative proteomics that are driving additional needs for both selectivity and sensitivity, and a lot of times without taking a trade-off. And as we talked about, earlier on, the trade-off between existing technologies really revolves around these 5 themes. It's sensitivity, selectivity, the overall development time of an assay, the reproducibility of that assay, and then at the end of the day, the cost. Not just the cost of the assay, but also the cost of the equipment that's required to be able to run it. And then lastly, we hope you've seen, with the advances in IMS and a lot of the supporting systems, there may be an opportunity now to address multiple trade-offs simultaneously, and again, overall, with the hopes of driving more success in gene therapy development and more of these molecules into the clinic. And with that, I will pass it back to Uduak for our audience.
Many thanks to our speakers for an informative and interesting presentation. We're going to begin the live Q&A session in just a few moments, so please submit your questions if you haven't done so already. Before we start the Q&A, a quick reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please do share the link with your colleagues who might not have been able to join us today.
And welcome everyone to the live Q&A. Thank you all so much for joining us. We hope you enjoyed the discussion so far. As a reminder, as was just stated, if you have a question, please feel free to enter it in the Ask a Question panel located on your screen. We have some time for questions, so please go ahead and get your questions in. Jason, Mark, David, thank you once again for that discussion. I'm going to jump right into the first question that I have here. Immune capture LC-MS hybrid assays is the technique that's often used to amplify sensitivity for LC-MS-MS-based methods. How does the approach compare to what you discussed on the IMS solution?
Udo, maybe I'll take that and then Dave, pass it off to you. But that's an interesting question. One of the solutions that I've seen us use here at Sonova is really understanding the actual protein and what peptides to target leveraging a high resolution mass spec and really seeing where you're getting the most intensity, and then shifting that over to an LC mass spec to really get to the sensitivity levels that you want. Recently, we went through an experience where we just weren't getting down to the sensitivity levels that were required, and we tried to go through an immune capture approach. And I think it really resonates on Mark's point and David's points with respect to, we struggled with the cross-reactivity or cross-selectivity of the antibodies, and then ultimately in just finding them and the selection and sourcing them. So, I think it's definitely something, a solution that's worked for others in the past, but it was one that really drove us to take a look at the IMS solution, frankly, at the end of the day. David?
Yeah, I think, one of the issues that you come across with immune capture, well, first of all, in immune capture, you have to develop the assay, which Mark, you have to develop the antibody, you have to find the antibody. It sometimes is hard to know what exactly is the epitopic region of a protein, right? So if you're looking for, a trans-species system, and you have a homology, there are a lot of homologies, let's say, between monkey and human proteins, you really don't know what epitope is binding. So you're starting off with some issue in selectivity, right? So you might be enriching the protein you don't want to see and missing the endogenous, the therapeutic sequence that you do want to see. So IMS, I think, ion mobility, because of its inherent selectivity, particularly in a bottom-up approach, but also in a top-down approach or middle-down approach, you will be able to manage that selectivity a lot better, and I think even it can reduce the development time because, again, you've added this extra tool of IMS, and you can certainly work a little bit faster.
Excellent. Excellent. Thank you both. Next question I have, can you provide more specifics on the trade-offs between immunoassay-based methods and mass spec-based methods, excuse me? And also, could you provide some additional thoughts on the trade-offs between the IMS solution and the traditional mass spec-based methods? I don't know, Mark, did you want to start? And then maybe David could jump in here.
Sure. So I think the trade-offs of using mass spec, as mentioned, was time to development and cost is just higher than immunoassay-based approaches. But at the same time, using some of these immunoassay approaches can come at a cost of sensitivity and specificity, potentially accuracy and precision. It just depends heavily on the antibody that you have, assuming you have one, and the protein you're trying to quantitate. Is it high abundance, low abundance? The similarity to other related proteins can impact specificity heavily. So what are you using the assay for? Is it fit for purpose? That's what you ultimately have to decide. And then if you're going to support development, then it needs to pass method validation. So it needs to have certain performance criteria. And that all depends on the science that you're trying to support. So it's very case by case.
Absolutely. Absolutely. And David and Jason, did either of you want to weigh in on this one too?
Go ahead, David.
Yeah, I think, I think the points that Mark made are the most relevant. The specificity of an IMS solution is really enormous. I mean, because by adding that extra parameter of shape, okay, you're really able to sort out from an array of possible interfering molecules. And again, to kind of repeat, you don't know where the epitope on an antibody is, and historically, people have done combined methods, right? Immune capture, you run your sample through some bead-based column, and you enrich your protein, and you can even do that for post-translational modifications. You can isolate phosphorylated proteins for stuff. But I think as the technique becomes more widely applied, IMS is actually going to speed things up, and it's going to also give you the key point, which is selectivity. I mean, particularly in gene therapy, that's selectivity is going to be very, very important.
Absolutely. Absolutely. Thank you. And just to remind our audience, we are taking questions live. We have a few more minutes. So if you do have a question, now is the time to go ahead and enter it in the Ask a Question panel on your screen. The next question I have, you mentioned some of the challenges with materials, so reagents and antibodies for immunoassay-based approaches. Could you provide some additional insight from your experience, and how does this impact the transition from the preclinical stage into the clinical stage?
Mark, you're the expert on that. You going to handle that one?
Yeah, sure. So, challenges with reagents, it all comes down to the antibody. You might not even have one commercially available. You have to generate one de novo. Even if you identify an antibody commercially, there could be commercial limitations to its use. It might be restricted to academia only and not for for-profit institutions, biotech companies, for instance. The issue that we had recently was the supplier just decided to discontinue the antibody. They didn't make any more. And so the qualification that we had with the antibody basically had to be thrown out, and we had to start again with a new antibody. I mean, there's ways around this if you secure licenses or some deal to secure future availability of the antibody if it's that important to support development. But you got to think about this not just for the primary antibody, but also the secondary antibody, the antibodies against the target that you're trying to detect, but potentially and likely a loading control protein as well. So there's a lot of things to consider, and a lot of these considerations honestly drove our choice to use mass spec, not just the specificity improvements.
Absolutely. Absolutely. Thank you for that. Next question I have, how do you see emerging technologies like AI-driven data analysis improving the accuracy of protein quantitation, both in traditional and IMS-based workflows? I'd like to take a stab at
I'll
that
start
one.
off.
Yeah.
I'll start off with that, Uduak, and then obviously hand it off to David and Mark for their thoughts as well. I guess it wouldn't be in the time that we're in a discussion if someone didn't ask a question about AI. But I think one of the things I've learned and I've witnessed on the new systems, in particular high res and what we're seeing on the IMS. But when on a recent analysis, we've seen tens of thousands of proteins and peptides in samples, right? And so there's just a tremendous amount of data available now. And I think I'm not the expert in what exactly you can do, but it's not lost on us that with that amount of data, some sort of AI approach or advanced data analysis is certainly going to be able to be applied to the proteomic space in totality. Frankly, I mean, I think recently you see the ex-head of Illumina, the traditional gene sequencing company, now joining Google. So I think coming back to the discussion we had earlier and that intersection of technologies, I think there's a lot of opportunity for AI, particularly in areas like IMS and high resolution, where the amount of data that's there and available, and even across different types of systems. I'm sure that AI will at some point in time have applicability to really amplify what we can learn from the data. Mark, David, any thoughts from your end?
Well, I would say computational tools are very, very important, particularly when you're looking at thousands of mass or even tens of thousands of mass signals for peptides from a digested or fragmented protein. All of the software that people use these days for analysis of the mass spec systems have, they're very, very powerful. And in fact, they're essential. However, you always have what we call the false discovery rate, right? So you can converge on the wrong answer. It's not common. But you in your software, you set these parameters. How much inaccuracy can you accept? And you can make that as precise as you want, but it is an issue, right? You can potentially see what you're not looking for, particularly. I mean, I've seen in our experience. I mean, the most famously occurs with leucine and isoleucine, right? Sometimes the software won't be able to distinguish them. IMS actually helps solve that problem for those particular amino acids. But you can focus on the wrong thing, and AI is a very powerful tool, and it is in fact involved in the interpretation of mass spec, not AI specifically, but something close to it. But at the end of the day, a human has to look at it, right? So the human being has to really correct the robot, if you will. It has to pass the Turing test, I guess.
Absolutely. And Mark, in the last few minutes, any thoughts on AI?
I use Microsoft Excel, so I'm probably not the best person.
Wonderful, wonderful. And on that note, I do have to bring the webinar to a close. Thank you all so much for being here. I appreciate it. And to our audience, thank you for tuning in. Before we go, one last reminder, this webinar will be available to view on demand in the next 24 to 48 hours. You should get an email with a link, and you can come back and watch the entire webinar all over again. And we also invite you to share the link with your colleagues who might not have been able to join us live today, but who you think would benefit from this discussion. So once again, thank you to all of our speakers for being here. I'd also like to thank today's sponsor, Sonova Analytical, for sponsoring today's event. And one last thank you to the Gen Multimedia Group for hosting. So thank you, David Mosley, Reynolds Gutierrez, and Natalie Torn. They make my job possible. And last but not least, thank you to the audience for being here and for watching us today. For everyone at Gen, I'm Uduak Thomas. Thank you and goodbye for now.